Today we're going to talk about virus structure. But before we do, I came across something I wanted to show you. Remember last week we were talking about the plaque assay. And for years I've always struggled to explain the plaque assay in a good way. Remember, we have a plate of HeLa cells, and each virus infection initiates the formation of a plaque. I explained how that happens. And I used this illustration to show that a virus first infects a single cell. The cell produces viruses which infect neighboring cells, and then they spread. Well, some movies were just published 2 weeks ago in Science. And I want to show these to you because these now, from this point on, I don't have to use this slide anymore. So what they did was they put a camera over on a microscope on a plate of cells as plaques were developing. So they did long-term infections, and they took a frame every so often and made a movie of it. So what you're going to see in the middle is where the virus infected, and the plaque is forming. So this is something that takes hours and hours or days depending on the virus. But you can see it in real time now. So that's the plaque. Right? We're just looping over and over. The virus starts in the middle. The cytopathic effect spreads outward, and you have a plaque. So that's what's going on in real time. Yes?
Why does it seem like that edge of the plaque is like having some kind of luminescence?
Looks like a stone dropped into a pond, right?
Yeah.
That's just as the refraction of the cells change as they're dying. They change their refractivity, so that's spreading out as the infection spreads. So that, I think, is the best way to illustrate a single plaque formation. Now, what they also did in this paper, they used a virus that has GFP, green fluorescent protein, in it. And that's the next slide. So now we're looking at the edge of a plaque. So here's the GFP spreading outwards. And here's that line of cell death you see again. And we're going to repeat it over and over. So here is the cells dying, spreading out, and then the virus, the GFP virus, is spreading as well. You can see it slowly is infecting new cells. Now, it turns out that this green fluorescent protein was expressed as a late protein during infection. So that means it's expressed late in infection. So you're actually seeing the cells dying before the protein, the green protein, is produced, which makes sense. The cells die first, and then the late proteins are produced. So I think that's the best explanation of a plaque I've ever seen. This happens to be vaccinia virus. And again, they just put a microscope with a camera and photographed it over the course of a couple of days. Just beautiful. Really nice. Okay, so today we're going to talk about the structure of viruses. We have a nice quote here by David Baltimore, a Nobel laureate. A virologist is amongst the luckiest of biologists because he can see into his chosen pet down to the details of all its molecules. The virologist sees how an extreme parasite functions using just the most fundamental aspects of behavior. So we know a lot of structural information for viruses, as you'll see today, probably more than any other organism. Viruses are like good buildings. So Walter Gropius, the founder of the Bauhaus movement, said, in order to create something that functions properly, a container, a chair, or a house, its essence has to be explored. For it should serve its purpose to perfection. It should be durable, inexpensive, and beautiful. And this is exactly how viruses are built. They are containers, and they serve their purpose perfectly, as I hope I'll show you in this lecture. Now, virion proteins have a number of functions. They protect the genome, of course. They help assemble a stable protective shell. They recognize the nucleic acid of the virion, so it gets put inside the shell. And they interact with host membranes for those viruses that have an envelope. The capsid proteins also deliver the genome into the cell, because after all, that's what has to happen in order for more virions to be made. So the capsid proteins will bind to cell receptors, as we'll see next time. They help transmit signals to get the genome out of the protective shell, a step that we call uncoding. And they help induce fusion with host cell membranes in the case of viruses with a membrane envelope around them. They also interact with components in the cell to help make sure that the genome gets to the right place. Depending on the virus, sometimes the genome has to go to the cytoplasm, other times it needs to get in the nucleus. and the capsid proteins help do that. And finally, the structural proteins also mediate other kinds of interactions with various cell components to make sure the infectious cycle is efficient, to transport components to the right place for assembly, so they have to interact with the transport machinery of the cell and with the host immune system, as we'll see a bit later. Now, one thing that I think is really important for you to take away from this is that virus particles are not inert structures. They are metastable structures, which means they have not yet attained their minimum free energy conformation. So let's explore this a little bit. A minimum free energy conformation of a virus particle usually is gained when an energy barrier is surmounted. And this usually occurs during attachment and entry, as we'll see. These kinds of energy barriers are surmounted and the minimum free energy conformation is attained. So really virus particles are machines, not just inert particles, that play an active role in the delivery of the genome. Metastability is the ability of a non-equilibrium state to persist for a very long time. Now, a good example of this is diamonds. So diamonds undergo very slow phase transformations at room temperature, and we say they are metastable because the transition to graphite, another phase, is extremely slow. So diamonds are metastable at room temperature, but if you raise the temperature significantly, the rate of phase transformation from diamond to graphite increases. It's no longer metastable, so it overcomes a barrier. Bonds in DNA and RNA and proteins are also metastable. They undergo phase transitions very slowly unless you provide energy or surmount an energy barrier. So for viruses, we can surmount this barrier following changes that happen during attachment and entry, as you'll see. So the energy barrier can be surmounted during that phase of the infectious cycle. In other words, in order to be infectious, a virus particle has to be metastable. The capsid has to protect the genome, in which case it has to be stable, but it also has to come apart quickly upon infection. And in that case, it has to be unstable. So the combination is metastability. So really, an infectious virion is a machine. It has moving parts, and it does work. Another way to look at it is that viruses are spring-loaded. During their assembly process, they're wound up like a spring. And then they will uncoat in the cell only if the cell provides the proper signals. So you put energy into the virion upon assembly, and then that potential energy, which is stored in the virus particle, is then used for disassembly. It's used to surmount the energy barrier towards the minimum free energy state. So viruses are really active containers. And in fact, for some viruses, not all, but for some, things happen inside the virion. It never completely comes apart. So replication and transcription of some viral genomes occurs while still inside the particle within the cell. The capsid opens up a bit. It doesn't completely release the genome. And examples are retroviruses, which never completely uncoat. The RNA is converted to DNA inside the partially uncoated virion, and reoviruses as well. The RNA synthesis, in particular mRNA synthesis, happens inside the virion. It never comes apart fully. So this isn't the case for all viruses. Some completely come apart, some extrude the RNA into the cytoplasm, but many are active containers. So today what we're going to talk about really is two distinct functions: structure and function. Structure, which is the particle itself, which we call the virion, of course. And the structure, as you're going to see, is created by the symmetrical arrangement of many identical proteins. So that's the key here. Viruses take a protein or a few proteins and repeat them many times to make a symmetrical particle that allows maximum contact between proteins, but they're not covalently bound to each other. They're non-covalently bound. The second part is function, which is genome delivery. The virus capsid has to deliver the genome into the cell, and this is made possible because the structure is not permanently bonded together. So don't taken out of your mind the idea that a virus particle is all covalently bound, because that's not the case. The proteins are non-covalently bound, but they're very stable because you have so many interactions repeated over and over. They're very extensive among proteins, and they're repeated over and over, and that makes for stability. But not complete stability, so the viral genome can get out. So upon infection, these capsids can be loosened or taken apart to release or expose the genome. So we have to have some definitions before we get going. The first is a subunit. The subunit is a single folded polypeptide chain. So when I talk about the subunit of a capsid, I'm talking about a single protein. Then we have a structural unit, and the synonyms for this are protomer or asymmetric unit. And this is the unit from which the capsids are built. And they usually consist of one or more subunits. It's the unit which, when multiplied, will give the intact capsid. The capsid, or the coat, is the virion itself. It comes from the Latin word capsa, meaning box. It's the protein shell surrounding the genome. Now there is another term which is a little confusing, nucleocapsid, or core. And this is the nucleic acid protein assembly within the virion. It's different from the capsid. And we'll see, I think, some illustrations of this which make it clear. Some viruses have an envelope or a viral membrane around them. That's a host cell-derived lipid bilayer. And finally, virion, of course, which is the infectious virus particle. So these are the terms I'm going to be using today. And this is one lecture where the textbook is very useful because there are a lot of embellishments in the book that will help you understand some of the concepts today. So this is all relatively new, the structure, and sometimes it's hard to get out of a single lecture. So I highly recommend you take a look at the chapter for this one. So remember in the beginning when I talked about what is a virus, I gave you some feeling of size. Let's do that again. Let's put things in perspective. A nanometer, of course, which we use a lot to talk about virus size, is 10 to the minus ninth meters. An atom is about 0.2 to 0.3 nanometers in diameter. The typical alpha helix in a protein is about a nanometer in diameter. DNA is about 2 nanometers in diameter. A ribosome is 20 nanometers. And poliovirus is 30 nanometers. And then among the biggest viruses are viruses, 750 nanometers. And I want to show you this lovely image which comes from the Protein Data Bank, which is a repository of all protein structures that have been solved at high resolution. And you can take the coordinates of all these proteins and make very nice pictures such as these. And these are a variety of molecules and enzymes. Here we have glucagon, for example, cytochrome C, and insulin. This is a cell membrane here with various membrane proteins in it. Here's rhinovirus, similar in size to poliovirus. And there's a ribosome to give you a sense of the size of polio versus ribosomes. And these are a variety of very large cellular enzymes. So you see many components in the cell approach the sizes of the smallest virus particles. So how do we determine virus structures? We have a set of 4 different tools that we'll talk about today. Electron microscopy, X-ray crystallography, cryo-electron microscopy, and nuclear magnetic resonance spectroscopy. And I'll just go over them briefly today. They're really well explained in the textbook chapter. I think that's chapter 4, not chapter 3. So a very important experiment happened at Berkeley, UC Berkeley in 1955, the first deconstruction and reconstitution of a virion. This really leads the way to modern studies on assembly of virus particles. And what was done here by Frankel, Conrad, and Williams, they mixed purified tobacco mosaic virus RNA and coat protein, and viruses formed spontaneously. So they started out with the virus particle. Remember TMV, the first virus discovered. They broke it up into RNA and protein. They purified each one. And then they took them and mixed them again, and they formed virus particles that were infectious for plants. And this was an extremely important experiment. because it showed that you could build a virus that was biologically active, it was infectious from the RNA and the protein component. And that got people very interested in studying the structure of viruses. And this virus, remember, not only was the first virus discovered, it was the first crystallized, first demonstration that RNA could serve as genetic material, and here the first reconstruction of a virion. The era of modern structural virology, I think, begins in 1940 with the development of the electron microscope to take pictures of virus particles. Here's the reference, and the one word I can understand is bacteriophage here. So he looked at pictures of bacteriophages in the electron microscope. You know, in the old days, when you got a PhD, you had to learn another language because papers were published in all sorts of languages. Now everything is published in English, so you don't have to take a language any longer. What's electron microscopy? Well, you take a microscope essentially, and instead of using light to view whatever you want to look at, you use electrons. So they have a higher resolution than light, so you can see smaller structures. The problem is, of course, that biological materials have to be stained for the most part. They don't have much contrast. So even if you're looking in a light microscope, you have to stain them unless you use other means. For electron microscopy of viruses, one of the common ways to look at them is negative staining with an electron-dense material such as uranyl acetate or phosphotungstate, and these scatter electrons. And this was something developed in 1959. The resolution of electron microscopy isn't great. You know, it's 50 to 75 angstroms. The diameter of an alpha helix is 10 angstroms, or 1 nanometer, so it's not great. So you can't get any detailed structure from this, but it allowed us to look at virus particles and get a sense of their overall morphology, and that was very important. So here's how negative staining works. You have a virion on a grid, and you put the negative stain on top, and when you bombard it with electrons, the negative stain repels it. And essentially what you're doing here is you're staining the background for the most part, and then you get a shadow of the particle, which you can then interpret. Okay, so negative staining stains the background to give you the detail. And when you put the stain on top of the particle, like it's done here on the left, you end up staining, end up having in the image both the top and the bottom of the particle. Okay, and as a consequence, you can be confused because you have images from the top and the bottom of a sphere, and that can be confusing. So that's this example in this electron micrograph here. But if you only stain half of the particle, say the lower half, then you get a better resolution of the real structure of the virion, which is shown here. The only problem with that is that it sometimes can make the particles collapse, so you don't have a good sense of their size. So we use negative staining of partial particles to get good images of what these viruses look like. And using this technique, you can take pictures of many, many different viruses. This is done till today. For example, you can find the electron micrographs of the H1N1 virus discovered last year on the CDC website. And this is, for example, a herpes virus, a hepatitis B virus, an adenovirus, a pox virus over here, all different shapes and conformations. And you can imagine when these were first looked at that virologists were amazed that there were so many different ways to build a virus particle. A more modern way to look at structure is cryo-electron microscopy. This is sort of like a CAT scan for viruses. What you do is you actually don't stain these now with uranyl acetate. You freeze preparations of viruses, and the freezing gives them enough contrast to see in the electron microscope. The idea is that in any field of virus particles, you'll have them oriented in every possible orientation so that if you could reconstruct all the images, you'd be able to reconstruct the particle. So you take a few hundred images of virions, you scan them individually, and then you do a lot of mathematics, including inverse Fourier transforms, to basically reconstruct the virus. So if you imagine each one of these is a virion in a different rotation, you can assemble all those rotations to make a 3D particle. The resolution isn't bad of this. It's about 8 to 20 angstroms, 0.8 to 2 nanometers. So you can get pretty good ideas of what viruses look like. You can't see typically the protein backbone, though. Here's an example of what you can do with cryo-electron microscopy. This is a cryo-EM reconstruction, it's called, of poliovirus in red bound to its cell receptor. So the virus was saturated with soluble receptor proteins, which had been purified, and the cryo-EM was done. Another thing that you can do with cryo-EMs, if you have higher resolution protein structures, such as those here, where you can see the alpha carbon backbone of the protein, you can try fitting that into the cryo-EM density. Now, structures such as these, high-resolution structures, are done by typically X-ray crystallography for large proteins. And that involves making a crystal of either the protein or the virus that you're interested in getting structural data from, and bombarding the crystal with X-rays. And the idea is that the atoms will reflect the X-rays based on their position in the crystal. So from the ways that these crystals diffract the X-ray beam, originally this was done using films, and you would get these circular diffraction patterns on films. It's now done by a detector directly into a computer. You can reconstruct the structure of proteins and even viruses. So for viruses, we can get easily 2 to 3 angstroms resolution and even less than 2 angstroms. So we can see basically all the amino acid backbone, and we can see the atoms as well. So the first to do X-ray crystallography, you need crystals. And in 1935, tobacco mosaic virus was the first virus to be crystallized, which means you make a concentrated solution of pure virus and you wait till it makes crystals. You actually have to see crystals physically. And he showed that these crystals were infectious, which was good. That meant whatever was making the crystals still retained its biological activity. And he got the Nobel Prize for that in 1946. And the first virus structure to be solved was, no, it wasn't tobacco mosaic virus. It was another plant virus, tomato bushy stunt virus, TBSV, which was done in 1978. And you may be wondering why plant viruses led the way early on. It's because it was easy to grow viruses in plants and get a lot of it to do these kinds of studies. The first animal viruses structure were solved was poliovirus and rhinovirus. Here's polio, 1985. The X-ray crystallographic study was solved. So we know where every atom is located in 3 dimensions. So if you go to the protein data bank, you can download a huge file which has the X, Y, and Z coordinate of every atom in the poliovirus particle, or at least every atom that was visible in the crystal. And then you could build a virus from that if you knew how. There are many programs now that you can use to take that data and make images such as this and get information about the structure. Now, you can see, I think, these lines here in this image. That is actually the alpha carbon backbone of the protein. So that's an X-ray crystallographic image. This is an image derived by cryo-electron microscopy. So you can see the resolution is much lower. You don't see the alpha carbon backbone of the protein, but you do get a sense of the overall shape of the particle. And sometimes this is all you can do for very large viruses. So how are particles built? So all of this these structural approaches have been used for a number of years now to get information on how viruses are built. And the principles are very interesting, and that's what I'd like you to take away today. The principles of building virions. Symmetry is the key to doing this. Now, Watson and Crick, you remember, very famous for discovering the structure of DNA in the 1950s, 1953, which I'll never forget that year. Happened to be my birthday. But they did more than discover DNA structure. In 1956, they pointed out that most viruses were either spherical or rod-shaped. So this is coming from a lot of electron microscopy which has been going on. And they were interested in this, and they noticed that most of the viruses look like either a rod or a sphere. They also knew that particles, virions, virus particles, are made with many copies of a few proteins. This was work that had been done in a number of laboratories. It was clear that a virus particle only had a few proteins in it. It was repeated, but there were only a few different kinds, or maybe even one kind of protein. And this makes a lot of sense, and it made a lot of sense to Watson and Crick because of what we call genetic economy. The viral genome is typically small. At least all the virions we knew back then, they were all small. So we said, well, you can't spend a lot of gene capacity on capsid proteins. So what you do is you take one or a few genes at the most and you repeat them many times to get a capsid. They also noted that viral proteins have structural properties that permit regular and repetitive interactions among them. So that's not true for all proteins. Not every protein will interact with someone of the same kind with repetitive and regular interactions well enough to form a capsid. But they noticed that this was a property of viral proteins. So they made an important contribution to virology besides discovering that DNA, the structure of DNA. They thought that how you make a virus is you take identical protein subunits, you repeat them many times, and you either use helical symmetry for rod-shaped viruses, or you use the symmetry of the Platonic polyhedra for round viruses. And Platonic solids, as you know, are icosahedrons and dodecahedrons and so forth. And they said this is how you put together round viruses. And they formulated the symmetry rules for viruses, which provide the rules for self-assembly. Rule 1, each subunit of the virus particle has identical bonding contacts with its neighbors. The repeated interaction of these surfaces leads to a symmetric arrangement. Remember, the proteins in a virus capsid are ideally suited to making these repetitive, extensive interactions with one another. That leads to a symmetric arrangement. Rule number 2, the bonding contacts between the subunits are usually non-covalent. And that allows for things to be taken apart. So the reversible formation leads to error-free assembly and minimizes the free energy. So if you make a mistake putting a virus together, you can take it apart and fix it. So in general, when you have identical bonds made between identical subunits, that gives you a regular structure. Now you may, if you've worked in the lab, you know that oftentimes you get aggregates or clumps of proteins, and this usually results when non-identical bonds form between the same protein. So you have two proteins and they interact in the same way, you will get a nice symmetrical rearrangement, a regular structure. If you have non-identical bonds throughout the proteins, you get clumps, aggregates, or precipitates or inclusion bodies. So that's one of the main differences between a nicely ordered virus particle and complexes that are disordered. The repetition of all these interactions results in a regular structure. And that is no better illustrated than in the fact that many capsid proteins, when expressed in a cell on their own, can assemble into a virus particle. So we call these virus-like particles. And for many viruses, you can make just one protein in a cell, just one viral protein. It assembles to form particles. And we have two vaccines, at least two vaccines today, that are virus-like particles made in yeast. The hepatitis B virus and the human papillomavirus vaccines are nothing more than a single polypeptide made in yeast. They assemble to form virus-like particles, and they are purified and injected into you. And the reason why they can self-assemble is because they have this property of being a capsid protein. They can make extensive and repetitive interactions, and they do so automatically. All the information to do that is programmed into the primary structure of the protein. So that's an EM of the HPV virus-like particles that are made in yeast, which constitute the HPV vaccine. So let's talk about the two different kinds of symmetry that are used to build virus particles, and we'll start with helical symmetry. This is very simple to understand. In helical symmetry, coat protein molecules engage in identical equivalent interactions with one another and with the viral RNA. So you get a very stable structure. So here, for example, is the RNA protein assembly of tobacco mosaic virus. So the green molecule here is the viral RNA, and that is bound by this protein subunit, the coat protein subunit. And many of these bind to the viral RNA. They not only bind to the viral RNA, but they interact with each other, and the result is this long helical structure. And this is, in fact, the virus of tobacco mosaic virus. So that's helical symmetry. These are about 300 nanometers in length. And in the electron micrograph, this is what the virus particles look like. Just the protein, the coat protein bound to the RNA in this helical fashion. So that's helical symmetry. There are viruses that infect animals that have the same symmetry. Here's one of them, Sendai virus. It's a virus related to measles and mumps viruses. It's constructed in the same way. You have a protein called the nucleoprotein NP, which interacts with the viral RNA, and the proteins also interact with one another. And they form this helix, which in the case of Sendai is about 1,000 nanometers long. Now, for animal viruses, these do not exist naked, as does the TMV and other plant viruses. These are usually encased in an envelope. And that's shown right here. So this is a paramyxovirus related to Sendai virus, and here's the envelope of the virus. This one is broken open, so the nucleocapsid is actually spilling out. So you can see this is this helical structure which is packed inside the envelope. So again, viruses of animals with helical symmetry are usually enveloped. Here's another example of a helical nucleocapsid vesicular stomatitis virus related to rabies virus. Again, a single protein, and repeated many times, it binds the RNA and itself and its neighbors. Again, repetition and extensive contact between the protein subunits makes for this very regular structure. And these nucleocapsids are also encased in an envelope such as this. So here is the VSV, vesicular stomatitis virus nucleocapsid, and it's encased in an envelope. So I'm using the term nucleocapsid to describe these two because they are the structure within the virion that contains the nucleic acid. Okay? Here I'm calling this the virus because that's all there is. It's not a nucleocapsid because it's the virus itself. This is the capsid of tuberculous virus. But for many other viruses where a nucleocapsid is packaged inside an envelope, we call it the nucleocapsid, not the capsid. So here are some more views of the rabies-like virus, vesicular stomatitis virus. This is an electron micrograph. You can't see the nucleocapsid within here because none of these have been penetrated by the electron-dense material. If you want to see the inside of these virus particles by electron microscopy, you have to depend on some of them leaking dye inside. None of that has happened here. But within these bullet-shaped enveloped virions are nucleocapsids with helical symmetry. So these are composed, again, of RNA bound to protein. And here's one subunit of this protein, the N, or nucleocapsid protein of vesicular stomatitis virus. And it binds RNA. Specifically, here is a structure of the protein with 9 nucleotides of RNA. And here is a structure of a number of these proteins on a circular RNA, just to show how the repetitive nature of the interactions leads to this helical shape. So envelope virions of RNA viruses with single-stranded RNA genomes often have this kind of structure, a nucleocapsid, a helical nucleocapsid with a membrane. Here are some examples: paramyxoviruses, measles and mumps, rabies virus, influenza virus, Ebola virus, just to name a few, just to give you a feel for what viruses are built this way. And again, the capsids inside the envelope are always called nucleocapsids. The nucleic acid protein assembly that is packaged within the virion, that's the nucleocapsid. And for these viruses, the nucleocapsid is not the virion. The virion is more than just the nucleocapsid. So how do you make a round protein, a round virus, from proteins that have irregular shapes? So now we're going to look at the structure of round viruses. We have a couple of clues before answering this. The first is that all round capsids have precise numbers of proteins, most often multiples of 60, sometimes others, but for the most part multiples of 60. So that gives us a clue. And secondly, spherical viruses come in many sizes. They can be 20 nanometers in diameter. They can be 750 nanometers in diameter. Yet, the capsid protein subunits that make up those particles are usually only about 20 to 60 kilodaltons in size. In other words, to make a really big virion, you don't use bigger and bigger proteins. So that's another clue about how to make a round virus. Well, way back in the early 60s, two structural virologists, Casper and Klug, were thinking about this problem, and this is the solution they came up with. They knew from Watson and Crick's work that round viruses are icosahedrons. This was determined after Watson and Crick published their first paper. Only icosahedrons are used to build spherical viruses, none of the other platonic solids. Secondly, capsid subunits tended to be arranged in the viruses, in these spherical viruses, as either hexamers or pentamers, groups of 6 or groups of 5. And you'll see exactly what that means. And then finally, the number of capsid subunits followed what they called were T numbers. You'll find out in a moment what T numbers are. And they were always precise multiples, 60, 180, 240, 960, and nothing in between. And so they devised the rules of icosahedral symmetry for building spherical viruses, because it turns out that an icosahedron is the most economical way to build a symmetrical shell with a maximum internal volume with proteins that aren't symmetrical in themselves. They're all different shapes. That's an icosahedron. An icosahedron, of course, is a solid. It has 20 faces, and each face is an equilateral triangle. The nature of an icosahedron is such that there are 12 vertices, and there are rotational axes of symmetry. I'll show you a picture of this in a moment. We have 5-fold, 3-fold, and 2-fold axes of rotational symmetry. This, again, allows the formation of a closed shell with the smallest number of subunits. You can take 60 copies of one protein, the same protein repeated over and over, and make an icosahedral shell. And there are many viruses that are constructed that way. And the icosahedron is the only way that virions with spherical virions are built. So here is a schematic of an icosahedron to show you how they're built. The 20 triangular faces, each an equilateral triangle, and the axes of symmetry that result. And all they mean is that, for example, around the 5-fold axis, which you can see here, and looking from the top, there are 5 of these faces or equilateral triangles. The 2-fold axis, there are 2 faces on either side of the axis. So here's the 2-fold axis, and here's the 3-fold axis. There are 3 equilateral triangles around that. It's simply a way of orienting ourselves in the structures of these different viruses. So let's look at a very simple icosahedral virus to establish these principles. And here is one made up of 60 of the same protein. So it's just one protein. It's drawn here as a comma. Some of them are orange and some of them are gray, but they're all the same protein. And you can take 60 of these and make an icosahedral structure, which makes a nice spherical virus particle. So here's the protein subunit. It also happens to be what's called the structural unit of this virus, because you repeat it 60 times to form the virus particle. All of these proteins interact with each other in an identical manner. They interact either head-to-head or tail-to-tail in the same way throughout the virion. And this is what Kasper and Klug first decided would be the structure of these simple viruses, and this turned out to be correct. So this is the simplest example, one protein repeated 60 times to make an icosahedral shell. So let's look at a real world example of that. This is a virus called adeno-associated virus type 2. It's a parvovirus, single-stranded DNA genome. It's about 25 nanometers in diameter, so it's a pretty small virion. It's made up of 60 copies of a single capsid protein. These T numbers we'll explain in a moment. So one capsid protein, and that's this one right here. We have the structure known. It's solved at X-ray crystallographic resolution, so we know all the positions of the atoms. And this is repeated 60 times, and that's the virion that's made by doing that. So again, you can see here the 5, the 2, and the 3-fold areas, rotational axes mapped onto the subunit, and also here. So for example, this blue region forms the 5-fold axis, and you can see 1, 2, 3, 4, 5 copies of that part of the protein around the 5-fold axis. So that makes perfect sense for a small virus, but we know that all viruses are not 25 nanometers in diameter. How do you make bigger viruses? 10 times, 1,000 times, 100 times bigger than this? You don't just make the proteins bigger, because we know that capsid proteins don't get bigger than a certain amount. So how do you do it? Well, you add more facets to the surface, and that's where this triangulation number T comes in. And again, I encourage you to go read the text if you don't understand what I'm telling you. The T number is simply the number of facets for a triangular face of an icosahedron. So this is the face of the icosahedron right here. Remember, there are 20 triangular faces. but you can put more proteins into the virion by adding facets to the triangular face. So let's see, here's a virus particle with T equals 1. There is one facet per face. So the face is the same as the facet here. This is made up of one protein. Here's a T equals 4 virus structure. We now have 1, 2, 3, 4 facets per triangular face. So this is the triangular face of the icosahedron. There are 20 of those in the particle. And we're adding facets to that to make it bigger. We're expanding the particle by just adding more bits to the facet itself. So you can see there are more subunits here than here, so the particle gets bigger. One of the consequences of doing this is now you have a new kind of interaction on the capsid. You have a 6-fold axis of symmetry. It's actually a pseudo 6-fold because in icosahedral symmetry there's no true 6-fold axis. But you now have subunits of the capsid interacting in groups of 6. Here we had them interacting in groups of 5. Here we now have both groups of 5 and groups of 6. So any capsid with a greater T number than 1 will have this 6-fold associated capsid proteins. So that's how you make a bigger capsid. Now this T number, this triangulation number, is rooted in mathematics that I don't feel you need to go into, but it's all explained in the text if you want to learn about it. It defines not only the number of facets per triangular face, but you can tell the total number of subunits in a particle by multiplying 60 times T. So again, the simplest assembly, T equals 1, there's 60 subunits. A T equals 3 icosahedron, you would have 180 subunits. T equals 4, 240, and so forth. So here are some examples of T numbers and viruses. So here the parvoviridae are T equals 1. And they have 60 subunits. The nodaviridae are T equals 3. They have 180 subunits and so forth. So for the most part, it follows the mathematical rules. There are some deviations from this, though. Some viruses do not contain the predicted number of subunits. So as we moved away from the original hypotheses of Casper and Klug, we realized that the viruses break the rules. So they don't always follow the strict rules of icosahedral assembly. So some viruses you would expect to have here 420 subunits, that's 60 times T, but they actually only have 360. So they break the rules. But the fundamental underpinnings still lie of what T number defines and how many subunits you calculate from it. Now the other consequence of having more than 60 subunits, when you start making bigger capsids, is that all the protein subunits don't interact with each other in exactly the same way anymore. They can't, because now you have groups of 5 and groups of 6, so there's no way they could all be interacting. They could be all in the same physical environment. So Casper and Klug recognized this, and they came up with this idea of quasi-equivalence. When a capsid has more than 60 subunits, each occupies a quasi-equivalent position. So in other words, the binding properties of subunits in different environments are similar, but they're not identical. Some have groups of 5, some have groups of 6, and there are other variations as well. So that's just, all that means is that they're not identical as they are in the T equals 1 60 subunit capsid. So here's an example of that. This is a T equals 3 capsid. You have 3 facets per triangular face of the icosahedron. And you have 3 modes of packing here, shown by orange, yellow, and purple colors here. You have groups of 5, and you have groups of 6. The yellows are groups of 6, the orange are groups of 5. So you have pentamers and hexamers. So they all have, there are some slightly different interactions in the capsid itself. They're not all the same as they are in the T equals 1. So that's all that quasi-equivalence means, and it's simply the consequence of making bigger virus particles. So here's an example of quasi-equivalence. It's a Notomura virus. This is a virus of insect, relatively small. T equals 3, which means it has 180 copies of a single protein, and this particle conforms to the T number rule. It does have 180 copies. And subunits, these subunits can interact in different ways depending on where they are in the particle. So for example, the B and C subunits right here are bent with respect to each other. They have a bent contact. But the B and C subunits here are flat. And we think they're flat because the RNA is binding to them, the RNA inside the capsid, and making it flat. But the point here is that you have different kinds of interactions among the proteins in the capsid in different areas, which is a consequence of making a bigger capsid. That's quasi-equivalence. Poliovirus is another example of one of these simple round viruses. It's called a pseudo T = 3 because normally a T = 3 would be the same subunit with 3 facets of the icosahedral face, and here there are 3 different proteins. The point that is good to make from the structure of polio is that the capsid proteins typically adopt this wedge-shaped structure. This is called an 8-stranded antiparallel beta-barrel motif. You can see there are 8 beta strands here, and it forms this beta barrel. And this is very good for packing together to form a virus particle. And it turns out that it's the motif most commonly found in virus capsids. Even though the protein sequence is very different, many of them look like this, so they can pack together to form a very stable structure. Here's another view of polio just to show you some real-life views. This is the outside of the capsid, showing each of the proteins colored differently: blue, yellow, and red. So you can see here very nicely a 5-fold axis of symmetry. This 5-pointed star, 1, 2, 3, 4, 5. And here's your 6-mer around here, which I had pseudo 6-fold axis of symmetry. And here's the capsid cut in half. So you can see it's got a relatively thin shell, and inside there is packed the viral genome. Now, a lot of these viruses are very corrugated. They have rough surfaces, as you can see from here and here. And we'll see that those are biologically relevant. They're receptor binding sites, and they're sites for binding of antibodies. Now, let's talk about a few more complex capsids, just to give you a flavor of how to make really big viruses. These include the largest viruses. They have distinct symmetries, but they also have extra proteins that don't fit into the symmetry, but have specialized roles in replication. So this is a table of all the structural proteins in adenovirus. Okay, there are a lot of them, and they vary in size. You can see none of them get over a certain mass. The exception is the hexon, which happens to be a trimer. And most of these are involved in constructing the icosahedral virion. But then there are accessory proteins that have additional roles. So, for example, there's a fiber that is involved in attachment to the host cell. There are a bunch of proteins that are called glue. They are thought to help stabilize the capsid, because now it's getting very big and it might fall apart. And then there's specialized proteins for getting the DNA genome into the capsid. Okay, so in addition to the basic hexons and pentons that build the icosahedral capsid, you have other proteins, and that's characteristic of these very big viruses. So here are some pictures of adenovirus. Here's an electron micrograph of the icosahedral shell. And one thing that you can see very notable are these spikes that extend from the capsid. And these are actually fibers that are part of the structure. So the virus is composed of 2 main capsid proteins, hexons and pentons. Most of the capsid is made of these hexons, shown here in light brown. And then at each 5-fold axis of symmetry, there's what's called a penton. It's a different protein. It's composed of a penton base and this fiber. And the fiber is important for attaching to cell receptors. So it's a specialized protein that's added to the icosahedral symmetry. And this is a molecular view of these proteins. This is 1 hexon. It's a trimer of this protein, so we know the structure of the hexon monomer. This is the hexon trimer, and each of these subunits here, these roundish shapes, is a hexon trimer. And then at each 5-fold axis, you have a different protein called the penton, which forms the 5-fold associated environment. And then attached to the penton is this fiber, which again is the protein that attaches to the cell receptor. Here's another example of a complex capsid. This is a real virus. Some of these viruses cause gastroenteritis. T = 13, 70 to 90 nanometers. It's composed of 2 concentric icosahedral shells. So when the structure of this was solved, it was very, very interesting, because the inner shell is a T = 2 icosahedron. And then the outer shell, which is surrounding this, is a T = 13. So 2 different triangulation numbers. yet they interact quite intimately. So this is the diagram of the real virion. Here's the outer shell in dark blue, the inner shell in pink, and then the RNAs are in the interior, of course. And at each five-fold axis of symmetry, there's a very interesting structure. As you can see here, it contains the sigma-1 protein, which is again used for attaching cell receptors. But at some point in infection, these sigma-1s leave, and this becomes a portal through which mRNA goes. So this virus makes mRNA inside the virion and ships it out into the cytoplasm through those portals. Yeah?
I have a different name for the outside and inside one besides. Because I saw, you said the matrix proteins or something?
That's different. Those are different viruses. I'll show you a matrix. Those are typically enveloped viruses. They have a protein below the membrane called the matrix, right? So you have membrane and then matrix protein. So these are pure icosahedral protein viruses, and they're called the outer and inner shells. Some viruses have capsids that are covered by membranes. These are called enveloped virions. They're very common, especially animal viruses. We don't know a lot of enveloped plant viruses. The envelope is derived from the host cell, and that's because the viral genome does not encode anything that can make lipids, as far as we know. So it has to get this from the host cell. And it's usually acquired by budding, which we'll talk about later, through a cellular membrane. It can be just about any cell membrane, although it is virus specific. So some viruses use the plasma membrane, some use the nuclear or Golgi. So any membrane will do, but it's always the same for that particular virus. The nucleocapsids inside this envelope can be helical. Remember the RNA wrapped in protein, or they can be icosahedral. They can be a beautiful icosahedral shell with an envelope around it. So this is an example of an enveloped virus. This is influenza virus. This virus has a membrane envelope with a helical nucleocapsid inside. Now, viruses that have envelopes usually have viral envelope glycoproteins in the envelope. Those are needed to be able to interact with the cell. So these are called integral membrane glycoproteins. They have attachment sites. They have antigenic sites. They have regions that are important for fusion, as we'll see. And here's a diagram of a typical one. Here's the part outside the virion membrane. There's usually a hydrophobic membrane spanning domain. And then there's a part of the protein in the interior of the virion that often interacts with other virion proteins. And these typically form oligomers. And so when you look at them in the electron microscope, they look like spikes. And the early EM people called them spikes. Here are two examples of these glycoproteins. Here's the glycoprotein of influenza virus, the hemagglutinin or the HA, which is standing at an angle to the cell membrane. And in contrast, there's another surface glycoprotein of flaviviruses, which lies flat on the membrane. They both have the same function. They attach to cell receptors, they mediate fusion, and this just shows that there are two ways structurally that you can accomplish this. So how would you put a flat glycoprotein onto a virion? Simply like this. This is a reconstruction of dengue virus with the glycoprotein of a different flavivirus because the dengue protein hadn't been solved yet. But you can see they're simply lying flat on the cell surface as opposed to sticking out as do the HA protein of influenza virus. Now these viral glycoproteins can interact with interior proteins in the virion. They could, here's the glycoprotein for one, interacting with, say, a capsid, an icosahedral capsid below it, which contains the nucleic acid, and hence it's the nucleocapsid. Or they can interact with a second protein. For many membrane or enveloped viruses, there's a protein just below the membrane called an M protein or a matrix protein. And these give structure and stability to the particle, and they often interact with glycoproteins, or there can be multiple protein layers. In most cases, these glycoproteins have some communication with the interior of the virion. So envelope viruses can be either structured or unstructured. So influenza virus is an example of an unstructured enveloped virus where if you look at it in the EM, it has no apparent symmetry. The spikes are simply embedded randomly in a membrane. This is an example of a structured envelope. So this is Synbis virus, which is a toga virus, an insect-borne virus. The capsid has T = 4 icosahedral symmetry. The capsid is surrounded by a lipid envelope which has glycoproteins in it. The glycoproteins interact with the capsid regularly, and so if you look at the virus from the outside at the glycoproteins, they look like they have icosahedral symmetry because they're binding with good interactions to the icosahedral capsid below. So for example, this is an image of the whole virion. So these are all the glycoproteins embedded in the membrane, and it has T = 4 icosahedral symmetry. And here is a cross-section of the virion. The RNA is in the middle. So this would be the nucleocapsid. Here is the membrane bilayer. Here are the glycoproteins embedded in the bilayer. And you can see the glycoproteins extend through the bilayer and contact the nucleocapsid protein below. So the nucleocapsid gives icosahedral symmetry to these surface glycoproteins. So here are some examples of unstructured envelope viruses: influenza virus, rabies virus, and some retroviruses. Retroviruses are enveloped, and in this case, the capsid is actually of icosahedral symmetry, but that is not passed on to the surface glycoproteins as for the Synbis virus that I just showed you. So we call these unstructured because when you look at them in the EM, you don't see any particular symmetry conferred by the underlying capsid. Herpes viruses are also large, complex viruses. Over half of the 80 viral genes encode structural proteins of the virions, 200 nanometers in diameter. There are 13 envelope proteins and 2 distinct internal structures. We have a icosahedral nucleocapsid, which has the DNA in it, and that's made up of 4 proteins. It's a T = 16 icosahedron. And then between the capsid and the envelope, we have what's called the tegument, a very complex structure made up of 20 different proteins, which has a role in the delivery of proteins needed early in infection. So it's very interesting that over half of the genetic information of this virus is devoted to making the carrier for the genome. Here's what the herpes particle looks like. It's a cryo-EM reconstruction. This is the exterior. So these are the glycoproteins embedded in the lipid, which is blue. If we cut away the virus to see the inside, here's the icosahedral nucleocapsid, and here's the tegument protein, the complex assembly of 20 proteins. These virions are icosahedral in symmetry, but they have one unusual feature that is inconsistent with icosahedral symmetry, and that is they have a portal, a hole basically in the capsid to let DNA in and out. And here the portal, you can see these very nice icosahedral nucleocapsids, and they're stained with an antibody to the portal protein. You can see the portal right there and there. And this portal has been reconstructed by imaging. You can see it here. So there's one portal in the whole particle, and it's at one of the 5-fold axes of symmetry. So it's sort of a violation of icosahedral rules, but this is what the big viruses do in order to adopt their capsids for specific functions. Other things can be in virions besides the virion structural proteins and the glycoproteins and the envelopes. Some viruses have enzymes, polymerases, integrases, other proteins associated with them. We'll talk about those in the life cycle stages. You can have proteases that are needed for processing the structural proteins, for cleaving them. Some viruses have an enzyme that makes poly-A. Put it on the RNA. Some viruses have capping enzymes. They put a 5-methyl G cap on the 5' end of the mRNA so it can be translated. Topoisomerases to unwind DNA. You can also have proteins that activate transcription that are involved in mRNA degradation and a variety of other aspects of infection. So these are all virus-specific proteins, but you can also have cell proteins in virions. You can have histones, tRNAs, myristate, lipid, cyclophilin A, and more. As we get more sensitive techniques for looking at virus structure, we realize there's more and more host proteins incorporated into virions. We used to think that an envelope virus didn't have anything in the envelope except the viral glycoproteins. But now we know there are other proteins derived from the host, and the question is whether they have any function or not. And we don't know pretty much the answer to that. Let me show you just a couple of examples of cell proteins incorporated into virus particles. So this is the structure of poliovirus, which I showed you earlier. The pseudo T equals 3 icosahedron. And this is the basic subunit. This is the protomer, or the asymmetric unit. It's composed of VP1, VP2, and VP3. So it's a T equals 3 virus. So you take 60 of these, put them together, and you make the virus particle. Embedded in VP1 of each subunit is a molecule shown here in white. It's a molecule of sphingosine, a fatty acid. It's derived from the cell. And there's 60 copies of that molecule in every virion, because one sphingosine binds to each protomer. This is the structure of poliovirus with a drug that it replaces the sphingosine in this pocket in the capsid. So that's that little yellow line here. And this is actually an antiviral drug that inhibits virus replication. And it does so by pushing out this lipid. And when we talk about virus entry into cells, you see the mechanism for that. So we think this lipid plays a role in uncoding. And by targeting it with antivirals, you can inhibit replication. So this lipid binds in a hydrophobic pocket in VP1, and that's shown here. And drugs were designed that fit very tightly in this pocket and push the lipid out. And these drugs stay there, and they block infectivity. So that's one example of a cellular host factor, in this case a lipid. Influenza viruses have another virus-coded protein in the virion, which is unusual. It's called the M2 protein, and it forms a channel. It forms an ion channel. So there are very few copies of the M2 protein in the virion compared to, say, the envelope glycoproteins. And these are allowing ions to get into the virion during infection. I'm going to talk about that specifically in the next segment. So here is a model of this ion. It's a very short protein. It forms a tetramer in the membrane of the virus, which is shown here. And during a specific part of the replication cycle, this channel allows ions to get into the particle. Now, you may have heard of the antiviral compounds amantadine or rimantadine, and those actually plug this channel. They fit right into that ion channel, and they block an essential step in infection, so they're inhibitory. Unfortunately, most of the seasonal strains of influenza and the pandemic strain are all resistant to this drug already, so it's quite useless. But when this was first discovered years ago, it was of some therapeutic value, and the target is this ion channel. Now, we haven't talked much about bacteriophages, but these are also complex viruses made up of a lot of the components that we've talked about so far. And in particular, I want to just briefly go over the tailed viruses. Most of the bacteriophages are tailed bacteriophages in the world, and they're composed of an icosahedral head. So this is built with the same symmetry rules as we've been talking about. You can see a blow-up of it here. And it's composed of a tail, which is attached at one of the 12 five-fold vertices of the capsid. And the tail is a very complex rod. It uses a helical symmetry, very similar to the symmetry we talked about of RNA viruses, protein-RNA interactions. And some of these, in fact, for different phages are contractile. And finally, it has long tail fibers, which it uses to attach to bacterial hosts. So the nucleic acid of these phages are stored in the head, and when these phages attach to a host, the DNA is injected through the tail and injected into the bacterial host. And here's a view of the base plate, which is composed of a variety of different proteins. And in many phages, there's actually a needle-like apparatus buried in here, which comes out, punctures the bacterial wall, and puts the DNA in. So a very, very complicated structurally virus, but very easy to solve. You just solve the components and put them all together. And I want to end up with the Mimi viruses, the biggest viruses that have been solved structurally so far. This has a T number of 1,179. So remember, the T is how many facets are in one triangular face. And I've been talking about T equals 1, T equals 3, 4. This is T equals 1,179. This is huge. So that's how you make a really big particle, right? You just put more facets in the icosahedron. So this structure was just solved last year by cryo-electron microscopy. And it's a big feat because these viruses have a covering of these fuzzy spike-like proteins sticking out. They had to get that off in order to solve the structure. And when they did, they found an icosahedral symmetry. But there's this star-shaped feature on here which violates all of the rules of icosahedral symmetry because it's only on one side of the particle. You see, when you turn this virion up, you see there's no star shape here or on the bottom. It's only on one side of the particle. Inside this particle is the DNA, which is shown here. It shows up as a compact region. And what is also interesting about this virion is that below the icosahedral shell, there's actually a lipid membrane around the DNA. So this is a reversal of everything I've told you. Usually you have a lipid envelope around a capsid. Here we have a capsid around a lipid envelope. We don't quite understand why that is yet. What's the star-shaped thing doing? The idea is that this actually opens up when the virus infects an amoeba, which is a host for this virus, and that's how the DNA gets out. So you can imagine this is a nice way to open up. Right? You have these 5 icosahedral faces just open up and the DNA can get out of it. That's a theory, it hasn't been proven, but it sounds pretty good. So these complex virions are really a challenge for structural biologists, and there are many of these to be solved. So if you like computers and math and viruses, all 3 of those things, you should become a viral structural biologist because there's always going to be plenty of work for you to do. And I'll leave you with this last slide, which is a wonderful website called ViperDB, and they have a really nice website where basically you can pick your virus, icosahedral virus, and build a picture of it. And all the viral structures, icosahedral viral structures are there, and you can pick your colors and views and make very nice images.